Hey guys, so this video is going to focus on the peripheral nervous system. So I wanted you all to recall how the nervous system is going to be organized. So moving away from the brain and spinal cord now to focus in on the sensory or afferent division. The peripheral nervous system also includes all of those motor outputs. So this is both the somatic or voluntary piece of the nervous system as well as the autonomic nervous system and its branches. So with the peripheral nervous system, you can think about it as allowing information to be passed to the brain and also a highway through which decisions that the brain makes can be carried out. So this is going to include all of the links from and to the outside world. To give us an overview for where I'm going in this video, I'm really going to focus on the sensory side of things here. So the levels of neural integration on the sensory side. So we have three levels. You've got the receptor level, the circuit level, and the perceptual level. When we talk about receptors, I'm going to briefly cover the different types of classification based on stimulus type. Then we'll look at this concept of adaptation and compare phasic receptors to tonic receptors. Then we'll look at the circuit level. So we'll circle back around to the thalamus here, as well as the reticular activating system. And then last up, the perceptual level. So this is at the conscious level. This is where we've reached the cortex. Because we're talking about perception, we're going to see that the cortex makes conscious interpretations of what it's perceiving. So whereas some people will first see the old lady, others will first see the young woman here. So this is the general pathway that we'll be following in this video here. So the receptor level, the circuit level, and lastly, the perceptual level. So you're seeing free nerve endings. So these are unencapsulated. You're seeing muscle spindles that help give us a sense for muscle stretch. This will come back into play when we look at reflexes. Then you've got kinesthetic receptors that you see concentrated at the joints. Let's take a look here first at the different receptor classifications. So you've got mechanoreceptors. So these are going to be responsible for sensing touch, pressure, vibrations, and stretch. So mechanoreceptors are going to include baroreceptors, which are going to be sensitive to those pressure changes. So blood pressure changes can be picked up both in the aortic arch as well as those carotid branches. Thermoreceptors, as you might imagine, are going to be sensitive to changes in temperature. So photoreceptors, such as rods and cones, are going to be sensitive to changes in light energy. And then chemoreceptors are going to respond to changes in chemicals. So this could be things like changes in pH. It could be chemoreceptors that are going to be sensitive to changes in oxygen or CO2 levels in the blood. And then lastly, the nociceptors are going to be sensitive to pain-causing stimulus. So extreme heat or cold, deep extreme pressure, responses or chemicals that are released during the inflammatory response are all nice examples here of nociceptors. So adaptation is a response that we see in these receptors, and it's a change in the sensitivity that's going to be due to the constant presence of a stimulus. So what you see here with adaptation is that a receptor membrane can become less responsive to a stimulus, even though the stimulus may still be present. And what happens as a consequence is that those action potentials are going to decline either in their frequency or they're going to stop altogether. So you've got two different types of adaptation. You've got phasic receptors that are phasic or fast. Both make that F sound. So they're going to be fast adapting is one way to remember it. So you're going to see that they signal the beginning or the end of the stimulus, but you are not going to have action potentials communicated as long as that stimulus is present. In contrast, tonic receptors will adapt slowly or not at all. Lots of examples of adaptation in our day-to-day lives. One example would be coming in from being outside on a bright day. Your eyes adjust to that bright light. And when you first come in, the room that you're in may first seem really dark. But your eyes, especially your rods, are going to quickly adjust and adapt to less intense light to allow you to be able to perceive your world. So what you're seeing here, you've got tonic receptors portrayed in this panel and phasic receptors portrayed in this panel. So with tonic receptors, remember that they are slow to adapt. So examples of tonic receptors are going to be nociceptors. So it's important that our body is able to perceive pain as long as it's present. In many situations, pain is going to be protective. Another example of a tonic receptor are going to be many of our proprioceptors that help to give us a sense of awareness, perception of stretch and range of motion in our joints. So with these receptors, we want to be aware of a particular stimulus as long as that stimulus is present. And so what we're seeing here is the stimulus begins, and this cell, this particular neuron, is going to begin to fire action potentials. And as long as that stimulus is present, those action potentials will continue. So with phasic receptors, remember that they are quick to adapt to a particular stimulus. So while the stimulus is going to be present during this whole period of time, this neuron, this receptor, is only going to be sending action potentials for the beginning of that stimulus. So fortunately for us, our sense of smell is going to be an example of a phasic receptor. So we may initially be very aware of the presence of a skunk in an area, but if we are to continue to stay in that area, then we are going to be less acutely aware of that smell, even though we are still going to be exposed to that stimulus. Another example, many of our touch receptors are also going to be phasic, are quick to adapt. So moving up from the receptor level to the circuit level, so recall that the thalamus is going to be primarily responsible for taking in all of the sensory input and routing that to the appropriate part of the cortex. And so when we're dealing with somatosensory input, our somatosensory cortex is going to interpret that. And we have our thalamus to thank for routing that information to the correct region. The thalamus would also be responsible for routing visual input to the occipital lobe, for routing auditory information to the temporal lobe. But again, we're just focusing on the somatosensory input here. In addition, don't forget the role of that reticular activating system. So this system very much acts as a gatekeeper to be able to determine what information is going to make it to our conscious perception here. You notice here in this graphic that some information, as it's routed up to the circuit level, is communicated to the cerebellum. And so because this information isn't going to the cortex, we're not actually going to be consciously aware of it. That's not to say that it's not going to be an important information that our brain will still interpret. It's just not being consciously perceived by our cortex. So now going up to the perceptual level. So one thing I want to do here is to draw a contrast between sensation and perception. So sensation is the awareness of changes in the internal and external environment, whereas perception is our conscious interpretation of those stimulus, which is really what I'm talking about here. So a sensation may be the awareness of deep pressure in your foot. And then maybe perception is the conscious interpretation of, I've got a pebble in my shoe, I need to take care of that. So just to give you one example of that. I really like this description here. So interpretation of the sensory input depends on the specific location of target neurons in our sensory cortex, and not on the nature of that message. So don't forget that an action potential is all or none. So another analogy that I like here is thinking about each sensory fiber as being like its own phone line to the brain. And it would be telling the brain not only who is calling, but from where that line is coming from, that line of communication is coming from. You may have experienced pressing on your eye and having your brain interpret that as a flash of light. And that's a nice example. So those photoreceptors, while they're being stimulated by pressure, the brain is still going to interpret it as a bit of light because it knows that it's coming from a particular neuron that is sensing visual input. So that's just one example here. This video link will take you to a neat video on synesthesia where we really see a disorder of this perception where people can sometimes hear colors or they will see tastes. And it's a really interesting crossover of this perceptual input. So with processing at the perceptual level, there are different ways of categorizing how our cortex is able to do this. First of all, perceptual detection is simply the ability to detect a stimulus in the first place. And we're typically going to see summation here where you're having multiple neurons communicate information. Magnitude estimation, as I've indicated a number of times, it's not the strength of an action potential, since an action potential is always going to be the same strength, but rather it's the frequency of an action potential, the frequency of those impulses that allows our brain to perceive a strong light versus a weak light, or a loud sound versus a quiet sound. Now, spatial discrimination is identifying the site or the pattern of stimulus. And a common way that this is tested in the laboratory is with a 2-point discrimination test. And so what you're seeing here is an indication of how far apart 2 points of stimulus must be before our cortex can interpret them as 2 inputs as opposed to 1. So you can actually use a caliper here where it'll start off really close together and then you can increasingly spread those points apart. And so at your fingertips, those points would not have to be spread very far apart in order for us to be able to tell that there are 2 points as opposed to 1 point. But if you have those calipers coming in on your thigh, they would have to be spread fairly far apart, on average about 70 millimeters, before you're actually able to interpret that they are 2 points present instead of 1. Whereas on our fingertips, on points on our face, those points may only be 2 or 3 millimeters apart before we can say, yes, there are actually 2 points of stimulus coming in here. And then lastly, pattern recognition. So our brain perceives a great deal in terms of patterns. So in very simple terms, we can take this pattern of colors and interpret it as a moon or as stars or as a Van Gogh. We can interpret patterns of sound as a particular melody as opposed to isolated units of note. So now I want to get into pain a little bit. So stimuli here, again, are going to include extreme pressure, extreme temperatures, and a variety of chemicals, many of which are going to be associated with the inflammatory response. So histamines, potassium, ATP, pH levels, bradykinins, those are all going to be things that are going to stimulate nociceptors. People oftentimes mix or confuse pain tolerance with pain threshold. So everybody has the same pain threshold, meaning that their nociceptors will fire action potentials at the same threshold of intensity. And that's going to be something that's consistent across individuals. Whereas pain tolerance is something that varies a great deal from person to person. So there are genetic components to this. There are experiential components to this, where simply being exposed to pain can change a person's perception of that. And so pain, we are really reliant on very subjective measures of pain, even this day and age. So, a very typical scale that may be given to a child would look something like this, where we are just reliant on how it is that they are tolerating that pain. And we will give them a scale from 0 to 5 or 0 to 10 to try to rate that pain. So visceral pain is going to be pain that is perceived coming from the thoracic or abdominal regions. So it tends to be pretty vague in nature. We tend to perceive it as a vague ache, a vague gnawing, a vague burning sensation. So phenomenon that are going to be adept at activating these nociceptors are going to include things like stretch, ischemia, so a loss of oxygen to a particular region, irritating chemicals, or muscle spasms. Those are common inputs that would trigger nociceptors from these visceral pain regions. So given that visceral pain can be so vague in its nature, referred pain can be really useful from a clinical standpoint. So it is the case that many of the visceral pain afferent fibers are actually shared along the same pathways as somatic pain fibers. And so a common one that many of you are familiar with here is this notion that a heart attack may be perceived by pain radiating down the left arm. And so the reason for that is you actually see fibers T1 through T4 that are shared between the heart. So those afferent inputs are going to be shared between the heart as well as those dermatomes along the left arm. And so that's why one of the warning signs of a heart attack can be felt along those motor pathways. And to elaborate on that a little bit more, what's really happening is because input is coming from both the visceral as well as the somatic, the brain really assumes that the stimulus is coming from the much more commonly stimulated somatic pathways. Other examples of this, gallbladder pain, also diaphragm pain is oftentimes felt along the shoulder. So you can see the diaphragm would be more along the left shoulder, whereas gallbladder pain may be felt along the right. Here is the referred pain from the heart and left arm. So some nice examples, but those referred pathways, again, can be useful from a clinical standpoint. So hopefully that was useful, guys. Thank you.